All right. Well, just a really quick reminder that your first exam will be coming up fairly soon, so a little more than a week. So again, those exams are open notes, open book. There is a study guide already on Canvas. I can show you that here. That'll work. So you can find the study guide right here in the Unit 1 material in Canvas. So go ahead, go through those. As a reminder, the ones that are basically a open-ended question, you can kind of guess that there will be multiple choice questions on those items. And then the rest of the exam is multiple choice questions, almost exactly like what you'll see on the exam. You'll see many, if not you'll see a lot of those same questions or same types of questions on the exam. So that should get you prepped and feel comfortable. I know students tend to be a little nervous for the first exam, but get studying now, review your notes, and you will do great. So any questions about that, the logistics of the exam? It'll be open from 12:01 a.m. on Friday, February 5th to 11:59, so just before midnight the next day, the Saturday the 6th. All right. Well, hopefully you are studying already, reviewing things that we've covered from chapters 1 and 2 as we're going through chapters 3 and then 4 as well. We will hopefully get started with chapter 4 today. If not, though, we'll start with chapter 4 on the 2nd and then finish it on the 4th. Chapter 4 is a little bit quicker than chapter 3, a little less material. It tends to be the way it is. Either the physical and cognitive development is the bigger chapter or the socio-emotional chapter in each age range is a little longer. So let's jump into it. Last time we ended up watching those videos of a human stem cell specializing into a neuron. And we also watched the video, maybe in this class we didn't watch the second video. I think we had time. So there was a second video, and it's very short, so I can just play it again. So the second video showing the plasticity of a neuron growing new dendrites. So this is vesicles, basically energy and stuff. But here you can see bits of the neuron, and here bits of the dendrites reaching out to make new connections. So when we're talking about plasticity, this is the process that we're talking about. The neuron actually expanding its dendrites or making new dendrites to connect to other neurons. So that was the final thing we talked about. And today we're going to start off with some other videos on how neurons actually send that message through the neuron. So a lot of times we use the analogy of the axon being like a wire, an insulated wire, but it's not actually sending a message in the same way an electrical wire does. So the electricity going to my light bulbs in here is just a steady stream of like electrons that go into the light bulb and make the filament or whatever it is hot and glow. That's not how a neuron works. A neuron is not shooting a signal down the tube of the axon. So it actually has a very different process that's a lot more efficient, a lot more effective, to be very fast. No matter how long the neuron is, it won't lose signal quality, which can happen with an actual electrical wire. It can lose some of its strength along the path with resistance and things like that. So it's actually a really interesting biochemical method to get a message down essentially a electrochemical tube. So the words I'm saying probably mean nothing to you if it was hard to understand in the chapter. So let's watch this video. It'll make it a lot more clear. I like its visual example, and we'll pause it a couple times throughout to talk about it. So let me know if you have any trouble seeing this video or hearing it as we go through.
Experience when you burn your hand results so quickly in an action by your muscles. Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body.
So before we jump into the detail of what's happening within that action potential, let's go back to this diagram. So hopefully you can see here, this sensory neuron is a neuron. So similar to the same interneurons that are in your brain. So this is an example of an interneuron, and those are the ones that are in your brain that are connecting, making memories, things like that. So the sensory neuron, interneuron, motor neuron, they're all very similar to each other, but they have different specific features. So this sensory neuron, the ends of it essentially can detect pressure, heat, light if it's a rod or cone cell. A hair cell also detects pressure changes. So, or a taste, for taste and smell, it's chemical. So the ending of those detects those specific things for a sensory neuron, and it sends that through the axon here to other neurons that in this case, especially when it's something like pulling your arm back from a stove, this message generally goes just straight back to your spinal cord and then straight to your muscles. So it goes back, the sense comes back to your spinal cord. Your spinal cord all by itself, just because of evolutionary processes, it knows, hey, to keep this body safe, I need to send that message immediately back to the motor neurons to be able to pull your hand back really quickly. So you will actually be pulling your hand back as your brain is now getting the message, oh, I just touched something hot. But your body all by itself is pulling that back. You can also stop that process like halfway through and then re-grab it, or consciously say, oh, this is going to be hot. So even if your body wants to pull back, you're like, oh, I'm holding my cup of coffee over my computer. I don't actually want to drop it right now. So you can stop that process. But this is a reflex, an automatic process that doesn't actually go through your brain to pull your arm back, which is kind of neat. All right, so let's go into the detail of what's going to happen at the molecular level, essentially, at the neuron. So again, this is a single cell, a single neuron. We're going to look at what's happening in an axon, like how it gets the message down the axon.
All neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of.
So an ion is just a charged particle. So whether it's a positively or negatively charged particle, this is definitely going to go into more detail chemistry-wise and biologically-wise than you need to know. But I think understanding this in this way will help you understand the basic processes a lot better. I guess another way to say that is you don't have to necessarily memorize every single one of these chemicals and the names of all these channels and voltage gates and things like that. If you understand a little bit of it, that's great, so that you can understand the more basic function of what's happening here. So don't be too worried about memorizing all of this.
These ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside.
So just at any general time, this is essentially, it's easier when I pull up like a whiteboard and draw on this. So let's say my arm is actually the axon. So these are the axon branches down there. So what we're looking at right now is basically a zoomed in portion right here on the outside of the top of this axon. So this is the inside of the axon, and then there's another side, another membrane like this on the other side of the axon further down here. And this is the outside of that axon. So fluid in the brain, you can see other neurons in the background. So there's other cells like holding neurons in place. So this is the space of the brain over here, and this is the inside of that axon. So to give you a sense of what it is we're looking at, where we're zoomed in. So if this is an axon, and this is the axon branches, we're looking basically at this little chunk right there. So hopefully that's a little more clear. And so the outside is just generally a little positively charged. The inside is just generally a little bit negatively charged.
The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential trend over the cell body. Multiple electrical potentials will combine at the axon hillock.
So this is happening so much faster than this video. This is happening in hundreds of thousands of seconds. We'll get to a slide that says exactly how many times a neuron can fire. I can't remember it just off the top of my head right now. But essentially, this is a really important process for building habits, for building memories, for doing actions. Because as you learn something new, you might be asking a neuron to do something it hasn't done before, to fire based on a new signal that it's never received before. So if you're trying to learn a new skateboard trick, it might take a lot of other neurons around it sending enough messages to this one cell to finally send that message down its axon. When you build something as a habit, though, let's say you've been skateboarding for many, many years. So maybe doing an ollie, just a little jump on your skateboard, is a habit, something you do basically instinctively. At that point, what that means is that this cell has gotten so used to firing from those signals, so it gets a signal from the previous neurons, and it's like, oh, I know, I've got to fire, I've got to send this message down. That's how habits are formed, essentially making it easier for this neuron to send that message. So when you're trying to break a habit, your neurons are used to firing in a specific way. They're used to sending the same like cyclical messages. Maybe you are primed or triggered by something in the environment. Let's take cigarette smoking for an example. So with cigarette smoking, you might have an addiction. You might smell cigarette smoke, taste something that you generally eat when you're smoking cigarettes, or drink as well. And that might trigger these networks of neurons because they're used to firing for that kind of thing. They're used to firing and saying, hey, time to smoke a cigarette. So when you're trying to break that habit, it can be really difficult because your brain is used to just firing and just saying, oh, time for a cigarette. So you're making new habits requiring these cells or other cells to do something different. And so that process of getting enough energy to fire that message down the axon can be difficult, which is why building new habits, building new memories works the same way. Essentially, while we're sleeping a lot of the time, some of our memories are like re-going over through their neural networks, and that's why it does it. So it makes it easier to remember that thing in the future. Reviewing information from your notes, it's basically making your neurons more used to firing for that information. Remembering, well, what is a sodium-potassium ion? So now you've heard that a couple times, and you'll hear it a few times more. So maybe by the end of this, you'll remember that a little better because it's going through those systems of neurons and they're getting used to firing and sending a message.
In a process called summation, if the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly.
So that's a really important point. The reason this process can be so fast is because it's not sending electrons, or it's not sending sodium potassium ions, it's not sending these things down the tube of the axon. Along the entire axon, it's essentially a wave. So at every different place, that little glowy light is basically supposed to be indicating where the action potential is along that process. So these gates have opened, these ones have not opened yet. So all that's happening is these molecules are moving very, very short distances. So this is the axon, the molecule, the charged particle moves in and out and in and out. So after it moves in, the inside of the chemical processes basically then push it back out to get ready for firing again. So that is why the process of neural transmission, the message can go so quickly, because it's not moving any of these particles all the way down, or like pumping it or pushing it all the way down the length of the axon. Some of your axons are up to 3 feet long, going from like the base of your brain down to about your tailbone. So that is a long distance if you were trying to push a couple particles. But if you do it in a wave where it's opening in a, oh, I think I did that the opposite direction. There we go. If it goes in a wave like that, it can happen a lot quicker. It keeps its same energy, it keeps its same strength the whole time, versus if it was like an electrical wire, it could potentially lose some of its energy along the way. So yeah, hopefully this is a lot more clear. It's basically a wave taking the positive and negative particles in and out of the axon.
Ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes.
So these are some of the axon branches. In this case, they're all basically touching the same cell, but this branch might be touching one neuron, this another, and this another, as an example.
To another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels.
So this is the next neuron in the chain of neurons, in that network of neurons. This is how it receives its messages. So these chemicals, let's say this is dopamine. Dopamine pouring out and coming to these channels. That is one example of a neurotransmitter.
Allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
All right, so hopefully you like learning about this topic even half as much as I like sharing about it, because it's really neat how those basic processes of the message going down a neuron and connecting with the other neurons are so central to the things that we do in life. So the things you think, the things you remember, your perceptions, your feelings of love or anger or happiness, They're all so dependent on this basic process of getting little molecules, ions, in and out of this axon and having the message sent and networked between a bunch of neurons. It's all really pretty neat, I think. With that being said, the myelin sheath, it does more than just insulate and kind of protect. It actually speeds up the speed of that that message. So up here, we have the beginning of a chain. Maybe you have a couple voltage gate channels to let the potassium ions in. I might be mixing those up, but that's okay. To let the ions in to start a message. So when it gets to the myelin, it can essentially skip this whole gap. So it has now changed the electrical polarization so that this whole area is at the same polarization as this one was. So it essentially kind of skips. So that's how it speeds up that signal transmission, is that you no longer need to open up all of these gates. You just have to open up these couple gates. And then you don't have to open up any of these. You just have to open up these. So myelination, as well as insulating the axons, can help speed up the transmission of messages. That's why it's not till about age 23, when the prefrontal cortex is fully myelinated, that we say the brain is finished developing, because that myelin is now all over, and so all the processing can be a little quicker. And we'll talk about in about adolescence how that actually affects behavior, risk-taking, and things like that. It's pretty neat. So questions about that process? Again, it did go into more detail than you need to know for the exam, but hopefully you understand that process a lot better now. So feel free to either raise your hand at any time, really, or type in a comment. All right, so going back to infancy. So the first years of life, there are major changes happening in the brain. So we saw the development of the brain structures before 9 months. Most things are there. From that myelination happens really rapidly, starting in the base area of the brain, like around here, and kind of moving in a wave towards the prefrontal cortex. Also, up until about age 2, you're making tons of new connections and then deleting any connections that you're not using. Again, you can make new connections at any times in your lives. But by about age 2, you've made about twice as many neural connections as you're ever going to use in your life. So you've made so many new connections, and then you'll start deleting the ones that aren't necessary and strengthening the ones that are necessary, and then making new ones as you go throughout your life, with that process of dendrites reaching out and touching new axons and other new pieces and new axon branches. So again, you can grow a whole new dendrite, you can grow new axon branches, but any neuron has only one axon. With that, each neuron is connected to as many as 1,000 other neurons. So the examples I've been giving are pretty simple, like one neuron talking to one other neuron. But one neuron can talk to as many as 1,000 other neurons. And so maybe some of those 1,000 neurons are part of the ability to do an ollie, versus some of those 1,000 neurons are part of another habit that you might have, or another skill that you might have. If you're playing piano, when you're first learning to play piano, part of what's happening, as well as learning to read music, learning to put where your hands are, your muscle memory, a lot of that is the sensory neurons literally learning and creating a memory of where to put your muscles, what position your hands should be in, things like that. So a lot of that is actually down to those neurons. So as you're learning a new skill, playing the piano, playing the guitar, you'll be using those neurons to develop that new habit. And as you do that, you'll be gaining new connections. So you'll be building new dendrites are going to reach out to make the process quicker and easier, so you can play more advanced music faster with more complexity, things like that. All right, so pruning is the name of the process of basically sucking in those dendrite arms. It's called pruning. It's not actually like cutting it off. It's just like reabsorbing it, but it's called pruning. All right, so let's jump into some brain structures. We'll kind of go through this pretty quickly. Your book does a good job going over these in a lot of detail. Some of the main ones that we talk about in the class are going to be the hippocampus, the hypothalamus, which I often confuse those two just because hippocampus and hypothalamus sound so similar. So if I'm ever doing that, I am sorry. I try to catch myself. And then the prefrontal cortex are probably the main ones throughout the course we're going to touch on, but all of them are obviously important. So hindbrain is the base part of your brain, back here in the lower part of your neck, or the upper part of your neck, the lower part of your head, where it connects to your spinal cord. It is the oldest part of the brain evolutionary wise. So if you look at lizards, they have most of their brain is basically hindbrain. And so it does a lot of basic survival functions like breathing, your heart beating. So that's all just taken charge of by this area of your brain. So the cerebellum controls motor, balance, so you're able to walk and things like that. Obviously, sensory neurons like in your ears are important for that too. So the brainstem is connecting the brain to the spinal cord. A lot of messages are being sent from here inside the brain and then down through that brainstem into your spinal cord into, say, your limbs and things like that. So with that, all of these are very important for basic survival skills. So most animals have them. So like I mentioned, lizards, if you compared a human brain to a lizard brain, you would see that this hindbrain structure is very, very similar, but we've got like a lot of upgrades, a lot of pieces added onto it, I guess you could say. So the midbrain is the very center, like the core part of your brain. Like if you, yeah, I think the picture does a pretty good job showing that. And so it's very important for connecting things that are happening up here in the brain to the lower part of the brain and sending new messages. So you are keeping your balance and you make a choice that, oh, I want to do an ollie. I don't know why skateboarding is so much on my mind right now, but it's a good example. So you want to do a jump on your skateboard. So you make that decision up here, and then it goes through this section of the brain, helps bring in the balance, and you do the trick. So with that, it's also got a lot of long axons connecting to many other parts of the brain, sending messages back and forth. The forebrain is usually what we think of when we're talking about brain, because it's like all the big wrinkly parts. So all the big outside shell is a big wrinkly mass where we have the limbic system, the thalamus, basal ganglia, cerebral cortex. So here you can see some of those. You can see the hindbrain, the midbrain, and then up here we're starting to get the limbic system and the thalamus, and then the cerebral cortex all the way out here. So let's go limbic system. This is where the amygdala and hippocampus are. Hippocampus is really important for memory. Amygdala is really important for things like emotions. So we will actually talk about the amygdala quite a lot throughout the class as well. How emotions, how memory, how prefrontal cortex and judgment all affect each other and how they work and interact. So if you think of this, you can think of this from an evolutionary perspective as well. It might help you understand like what the different components of the brain do. I mentioned the hindbrain being very similar to a lizard's brain. That's basically what a lizard has. And then think of other creatures that are maybe more similar to us. So like apes have limbic systems as well. Apes also have cerebral cortexes, but not as developed of a prefrontal cortex. So you can kind of look at what animals are capable of and think of, oh, well, maybe this part of the brain is responsible for some of those things. I don't know. It's one example, one connection that helps me remember, like, as you kind of move outside in the brain, it's basically more highly evolved or capable of a lot of the things we as humans are capable of. All right, so the thalamus is also a relay station and integration of information. So this is an actual picture of a brain right here. So you can see how it's connected to the midbrain there, and you're bringing in information from the cerebral cortex. So with all of these, they have axons sent to each other. This is actually the corpus callosum right here, and it's all it is axons that are going from your right and left hemisphere of your brain. So it's just a ton of axons. If you ever get the chance to dissect a brain, I think it's interesting to poke it versus poke this area of the brain, because this is very hard and dense because of how tightly packed the axons are. There's so many there to make connections between the hemispheres of the brain, versus this is a lot more like mushy. Sounds really gross as I say it, but yeah. Anyway, so there's some of these that I'm just going to kind of skip over right now. So hypothalamus, it is not the only pleasure center in the brain, but it is one. The hypothalamus is very central in a lot of regulation of the body's internal states. So reward, punishment, eating, drinking, sex, a lot of those things are going to make the hypothalamus more or less active and help with motivation. So it is an important piece. Cerebral cortex is, if you opened up someone's skull and looked at their brain, essentially what you would see is cerebral cortex, the outer wrinkly part. That's where a lot of different things are going on. Temporal lobe, a lot of that is involved in hearing. Occipital lobe, a lot of that is involved in seeing. Somatosensory cortex, that's where the messages are sent of, if I pinch myself on my wrist, I feel that, and that message has gone to the somatosensory cortex. If I want to move my fingers in a specific way, like trying to do the little wave with my fingers I did earlier, that's going through the primary motor cortex. And then frontal lobe is a lot of our higher functions. And then prefrontal cortex is where like judgment comes in as well. All right, so, I just went through all this. So one way that we learned about this is from looking at humans with, say, injuries before MRIs even existed. So some of the ways we first started to realize that certain areas of the brain were responsible for different things were from people who had injuries, whether it was a disease that after dissecting, after the person was dead, or in this case, Phineas Gage, we could learn a lot about the frontal lobes. So does anybody remember the story of Phineas Gage? What is he holding here? What's going on with his eye? Why is he on this slide at all? Anyone with a microphone, go for it.
I barely remember. I think something got into his eye and up to his brain, and they were able to take it out, and he survived. But then he changed all his emotions and the way he thinks. He was not the same person as he used to be. Something like that.
Yeah, exactly. You hit all the most important points there. So this is a railroad tie, basically a really big nail you could think of to hold railroad beams down. So he was working on a railroad and it exploded and it went into his skull. So I believe it went into his skull about here, so it did damage his eye, and it went through his frontal lobe. So just like you said, he did survive. They were able to, he was able to survive that injury and even collect the tie, the railroad tie. But just like you said, his personality to the people around him, his personality completely changed. Beforehand, he was a pretty nice guy, just generally friendly. But after, he was very emotionally shallow. He was very distractible. He didn't want to follow directions. People would say he was rude. So his personality changed dramatically. And so what they realized was, well, maybe the frontal lobe, had the part of his brain that was damaged, was responsible for some of that behavior. And so over time, we've been able to realize from like that and other injuries and then MRIs more recently, the parts of the brain that are responsible for a lot of those things, or partially responsible. Because again, your frontal lobe connects to other areas of your brain. It's not the only part responsible for you being nice. Also, you've got the midbrain and your emotions talking to this part of your brain and then making decisions, sending that back. Yeah, really great memory of what happened to Phineas Gage. I don't remember if this class I have it. In my other class, I have a picture of his skull after he died, and it still has the hole through it, which is pretty cool, and you can see exactly where it is. But I guess that's not in these slides. So the cerebral cortex in infancy is in the process of learning how to do things. So your temporal lobe, where a lot of your hearing goes to and is processed, it's starting to work a lot even in the womb. So you're starting to get used to the sounds, like the volume level, the pitch, the rhythm of human speech. You're starting to get used to that even in the womb. So it's getting some use, but the occipital lobe isn't doing too much because it's pretty dark in the womb. You've got maybe a sensation of light versus dark, but that's about it. So once a person is born, these areas of the brain can do a lot more, and are basically learning what they're responsible for, how to do their job. So with that being said, you can look here at the level of basically dendrites and axon branches in each section of the brain. And you can see that they start to prune a lot. So here the visual cortex starts pruning really early, so about age 1. So you get to about 20/20 vision, you have that for a couple months, and then you start pruning a lot of the connections that you don't need anymore. For auditory, you're learning language in those first couple of years of life, and then you start pruning all of those away. Then prefrontal cortex, this is still, obviously these aren't like a continuous decrease of a line. They stay about this level, and if you watched, it would just kind of wiggle this way. It's not like all of these are decreasing the whole rest of your life. Yeah. So with that, the prefrontal cortex, I've mentioned it quite a few times. Hopefully these are things that you remember about it because it will come up many times in the class. So higher cognitive functions, your planning, your reasoning, your self-control. So if you're trying to plan a trip to Arches National Park, you're going to be using your prefrontal cortex to do that. So think ahead of what you're going to want to do, maybe have some self-control so that you can pack food instead of spending all your money on crappy road food or something like that. So it also has your executive control system where you're doing that in an immediate time, not just like planning a trip later on. So somatosensory cortex is where your messages of the things you're feeling around your body go to. And so basically these pictures are essentially just indicating how much mass of neurons are dedicated to each part of our body. So there's a lot of neurons dedicated to the mouth, including like the tongue and the lips. And there's a lot of sensory neurons in our brain dedicated to the hands. You can see there's not very many for the trunk or the legs, but there are significantly more for the genitals. So you can also see that here in how many are focused on neck, eyes, and nose, genitals, versus here we've got toes, feet, legs, and trunk, the whole head over here. So there's a lot of space dedicated to the senses that are going to be required for fine motor skills in your hands, for hearing, for lips, things like that, taste. And this is some of the reasons that children often put things in their mouths, because they do have a lot of sensory nerves there. It's going to tell them a lot more about an object than, let's say, putting it in their elbow. They'll have some sense there, but obviously putting it in their mouth, they can tell texture a lot better than you might be able to in your elbow. Don't know why I use that as an example, but there it is. So motor cortex is essentially right next to the sensory cortex, and very similarly, there's a lot of space for moving your tongue, which is also important for speaking. There's a lot of space for motor neuron, or these are interneurons sending the message to the motor neurons, but there's a lot of interneurons dedicated to the movement of hands, versus over here, there's not a lot dedicated to movement of toes. Now, you might have seen videos of people who maybe they've lost their entire upper limbs, or maybe they've lost their hands and they've learned to maybe do art or play an instrument with their toes. So what do you think is happening there? So right here in the average person, this is about the space that's taken up by the hand versus this is about how much is taken up by the toes. So what do you think is happening at the neural level to give the people that ability to be able to use their toes so with such dexterity, to be able to play the piano or another instrument or draw with their feet and toes. Yeah, very good. So we've got a comment in the chat. Hopefully you're also watching the chat at the same time I am as well. So the hand area is decreasing and the toe area expands. So once these neurons realize, hey, I'm not being used, or I don't need to be used to use hands. If a person's missing hands, they're not going to be sending those kinds of signals much anymore. So this area for the toes, it can do it in one of two ways, either expanding, like this area takes up more of the neurons here, or some of the responsibility for this area is actually taken up by this. So these neurons might now repurpose themselves to do the things, rather than working with hands, they might work with the toes now. And that's just dependent on the connections between them. So, and so for any given person, it might be different. This area right here might expand. More of this is gaining more connections, being more dense, versus this, it's switching responsibility over. So there's a couple different ways the brain can do that, but it's pretty neat. So that's a really good question, and I was actually wondering if I should mention it right now. So what's happening with the brain and phantom limbs, like when people who have lost a hand feel that hand? So that goes a lot back to the previous slide of the somatosensory cortex. So those neurons, those interneurons in your brain, well, it can be one of a couple things. One of them is these interneurons in your brain, they're still part of networks of when you feel things. So let's say if you pinch your hand right here, you might even also feel like pain in your arm right there. So if you at some point, let's say you've only lost like this part of your limb, that same pain right here, after losing that hand, you might still feel some pain or sensation there because of the neurons cross connections, because of the networks that you're activating. That's one thing that can cause that phantom limb pain. Another thing is your neurons going down here are the sensory neurons are part of networks themselves. So even though you might have lost your hand, you might still have some sensory neurons here that are part of the network sending the message to this part of your brain. So if it's still sending the message to the part of your brain that can feel your hand, you might still feel a pain in your hand, even though in reality you don't have a hand there, but you're still getting that message of pain in your brain to your hand. So that's a couple different ways that can happen, which is unfortunate for a person who no longer has that hand to then also have that pain for that. But it is interesting how the brain is working to do that. So really good question. So I mentioned corpus callosum earlier. It's basically a bunch of axons going to both sides of the brain. Has anyone ever heard of whether they were right-brained or left-brained in the class? Let's just get if you've heard of it, and you've ever, say, done a test, or you know what right brain versus left brain should be, just put it in the chat really quick if you think or you've been told that you're right or left brained. All right, we've got a couple comments coming in. So far, they're all left-brained. So would one person be willing to share maybe on the microphone? And some people haven't been told one way or the other, which is just fine. So for those of you who have heard or have taken a test to say you're left-brained, can you remember what left-brain people are supposed to be like or do? What is left-brained, I guess, is my question.
I was not told if I was left brain or right brain, so I'm not sure if I'm saying this right, but I think left brain should be more analytical, more like into mathematics and thinking in a more analytical way, and the right side should be more like creative. I don't know if it's right or not. I have no idea.
No, that's perfect. Thank you. I often forget too which one is supposed to be right versus which one's supposed to be left. But you're exactly right on the differentiation. When people say, "Oh, I'm more right brain or left brain," they're either talking about they're more analytical or they're more maybe creative and possibly artistic. So we're learning about the corpus callosum here. That's essentially a large part of your brain that's sending information from both sides of your brain. So I guess what I'm trying to do is ask, well, how does that idea of left versus right brain fit in with knowing now that you've got essentially hundreds of thousands or maybe probably millions of connections between your right and left brain? I guess basically I'm trying to make a little bit of cognitive dissonance here, holding two different ideas at the same time, basically, because really there is no such thing as right brain versus left brain. Now, it very well might be the case that you are a little more analytical versus a little more creative, but that's not actually down to either the left or right hemisphere of your brain. So the left and right hemispheres of your brain basically work for your left side of your body and right side of your body. So the somatosensory cortex on the left side of your brain is feeling the things that are happening on the right side of your body. So that's the main difference between the left and right hemispheres of the brain, is that the left hemisphere is responsible for things that are happening on the right side of the body versus the right hemisphere is responsible for the things happening on the left side of your body. I'm pointing to sides of my head and I'm hoping that I reversed the video so that it actually makes sense as you're watching it. Yeah, right side of your brain is responsible for feeling the feelings in your left hand and sending a message to move your left hand, and vice versa. The things that we call personality traits, like creativity or being more analytical, that takes so much of your entire brain, not just focus. Some aspects of it might be slightly more focused in one side or the other. So processing language is more focused on the left side of our brain, like right about here. So most people are generally processing language there, but for almost every other thing, we're using this corpus callosum. We are using so much of our brain, not just one little segment of it, but all of it together in a network, that it's not that being right brain versus left brain as an actual location in your brain isn't actually a thing, which means it's totally fine if you've not been told one way or the other you're right brain or left brain. Nobody is right brain or left brain. I guess I shouldn't say that. Some people do actually have had a whole hemisphere of their brain removed. I wasn't planning to go into this, but that's fine. So if someone has seizures that are only starting in one area of their brain, one treatment for that is essentially to kill the neurons in that area of the brain that are starting the seizures. So that's a lot more focused. You can do that with maybe an electrode put into the brain, kill a couple neurons where the seizures just tend to start from. But if someone's really, really bad and their symptoms are really bad, especially if they're younger, so this is usually done closer to childhood, there have been patients who have had a whole half of their brain removed because of all the problems that it's causing the rest of them. And those people still, especially younger, remember plasticity. So that one half of the brain is the only one that's there anymore, and it can do all the same things that having two halves of their brain can do, which is pretty neat. Obviously, it needs to take some time to learn to do all those processes. And that's why it works better for doing that for a very young person versus if you took half of the brain of someone who's in their 30s, they're going to have a lot harder time rebuilding some of their skills and abilities, or strokes as well. That can also basically delete a big portion of your brain, but because of plasticity and that axons and dendrites, it can Not heal itself. It's not actually healing the dead neurons, but repurposing other neurons and making new connections to take over those responsibilities. I see a question, so go for it.
It's not actually a question, it's just a comment. I watched like a video of people that had strokes and they went to therapy, like physical therapy, to start like gaining movement or the where they lost.
Mm-hmm.
So on the therapy, they didn't let them use the hand that worked or the side that worked. They only let them they put like like those gloves that you put like to touch the hot oven. So they put that on the hands that worked, so they don't have like much movement that they can grab things. It makes more difficult. So they have to use the hand that didn't work. And at first, most of them couldn't do anything, but they kept trying. So with like within months, they all were able to regain all the movements again from the side that was not working, because they were not allowed to use the side that worked.
Yeah, that's a fantastic. Yeah, that's a fantastic example. If you remember what it is well enough, if you could send me a link to that, if I can find a short clip, that might be something I would actually show to the class in later semesters. If you can't find it, no problem. But that's a fantastic example. So what is happening there in your example is that, let's say this part for your right hand is damaged. So you're no longer able, because of stroke or injury, something like that. Maybe it got injured playing football. I don't know. So just as an example, there's a lot of head injuries in football. So you can no longer use your right hand as you used to be able to. So what is happening in that process of essentially forcing a person to use the hand that's not working is the other neurons either are around there, part of that network that were part of the network, are now becoming more responsible and learning and making new connections, sending out new axon branches, sending out new dendrites to be able to do that process of now moving that right hand again. Or it could also be the other half of the brain, the other hemisphere is taking on that responsibility, and so pulling double duty. Rather than just doing the left hand, it's also doing the right hand at the same time. So that process of forcing a person in a therapeutic way like that, in a focused way, like step by step, doing specific tasks, is retraining their brain to develop those new axons and dendrites to be able to do that task. And depending on what it is and depending how extensive the damage is, will depend on how well or not well a person can recoup that entire capacity. So if someone was able to type at, I don't know, let's say something crazy like 400 words a minute, that's a lot higher than I type. I don't know if that's actually a crazy speed to type. They might not ever get all the way up to 400 words a minute. Maybe they're only at like 200. But that's all really dependent on an individual, where their injury is, how extensive it is, and also taking proper vitamins and nutrients so the cells can grow those new axons and dendrites. But that's a fantastic example. Thank you for sharing that. It's amazing to think of how many things are really dependent on those neurons. All right, so really quickly, let's talk about the endocrine system. So it is part of your nervous system. It's a system of glands. I mentioned this last time. And so it is connected to your brain through the pineal gland, through the pituitary gland, through the hypothalamus that is sending and receiving. So the pineal gland and the pituitary gland are creating hormones, and then other aspects of your brain are sensing those hormones in your body. But we also have thyroids, thalamus, ovaries, pancreas, adrenal glands, testicles, placentas, and those all create these different hormones. And it's part of your nervous system. So it's part of the body's processing of sending and receiving messages back and forth. The hormones in the endocrine system tend to be a lot slower. So the neurons, they're using that cellular process to that wave processed down an axon to send that electrochemical message really quickly. For the hormones, we essentially have to get to a specific level of the hormone in our bloodstream before other organs start to recognize that that message is being sent, or the areas of the brain start to recognize. So you're kind of dependent on the speed of the blood flowing, essentially. So it's a lot slower process, and it has to build up, versus an action potential in a neuron is just really, really quick. All right, so this, going back to infancy, the changes in the brain occur because of experience. So especially for young children, play is just so important. So that's in infancy as well as up into the teen years. So you don't, especially for young infants, you don't need special toys or special music. They just need a variety of experiences. So different colored blocks of different shapes and textures is a fantastic toy for children. So they can feel things that are smooth, things that are rough, things that are hard versus soft, see different colors, feel different textures. I might have said that already. But basically, for a child to start to grow and learn and build skills like motor skills, fine and gross motor skills, they need to have experiences. They need to experience the world. So it's a big part of the brain developing and making those connections. Yeah, if you want a child to be really good at building Lego, give them Legos early on. Well, maybe the big ones so they don't swallow it and die. That would not be good. Give them Lego early on. That's a really good example of a block, or wood blocks. You don't need a bunch of really special, specific toys. I see a lot of toys sometimes marketed like, this will be really great for your developing infant. It has all of this variety of things, and it looks really complicated and maybe really impressive to an adult looking at that, like, oh, they could use this for this and this for this. Simple variety is really the best for infants. All right, so infant sleep. Newborns sleep on average 16 to 17 hours a day. Their parents probably a lot less than that. But with that, their sleep is in little chunks. So newborn sleep sessions are about 3.5 hours long. And then they get to be about 10.5 hours long from 3 to 7 months. So any individual child's going to be a little different on that. So my children kept that same about 2 to 4 hour sleep schedule about to 6 months. So any individual child's going to be a little different. But generally the sleep is in those little chunks. And then the time in between sleep sessions is generally a restful wakefulness. So there might only be a half an hour that you can really actually play with a newborn and have them interact with you in between their sleep sessions. So taking advantage of that is really good. And then also as a parent, taking advantage of that time when they're actually asleep and sleeping is good. Obviously, if you have to go back to work immediately and you don't have maternity or paternity leave, it can be really difficult to do that. So, yeah. With that, SIDS is a risk. It's a highest risk at 2 to 4 months and much less likely after 8 months. Essentially, this is when the infant stops breathing during the night without an apparent cause. Sometimes it can be pointed to things in the bed with them. Maybe they're on too soft of a cushion or a mattress, or there's a blanket in there and they got their face in the blanket. So it's best to have, especially really young newborns, in a fairly hard mattress. So not like a hard metal plank or something like that, or wood plank. I guess you wouldn't have a metal plank, but something that is more firm than you would suspect. Because we often think of we want to cradle and cuddle the baby. We want them to be on soft clouds as they sleep or something like that. Well, it could make them die. So let's not do that. So hopefully your pediatrician will give you advice on that specifically as well. Yeah. So with that, place babies on their back to sleep, not their side or not their front. It's easier for them to breathe well and not overheat. Bed sharing is also a higher risk factor, and this is a hard one because for the parent and the child to share the same bed can make it a lot easier to do feedings over the night. It can increase that attachment between the parent and the child and that bond there. But the parent will probably want to have, I don't know, a blanket or a pillow, whereas it's not as safe for the child. So also smothering the child is also a risk factor there. So there are beds that can like attach to the side of beds. So it does make it easy to, say, feed the child, but they're kind of in their own space. Those are probably some of the better options for bed sharing, but obviously it's a risk factor. It's not any, you've got to weigh as a parent the risk you're going to put the baby in, obviously. So yeah. This is an argument that, especially when we were going to have children and some of our friends were going to have children, it seems like was an argument that was happening a lot, which is funny because science has done a lot of looking at breastfeeding versus bottle feeding. Breastfeeding does seem to be a little bit better, even than modern day formulas. Say decades ago, breastfeeding was definitely better than bottle feeding. For the nutrients that the child is getting and so on and so forth. Now the formulas are quite a bit better, but breastfeeding still does seem to have some advantages, but not to the point to say we should shame anyone for not breastfeeding, especially when some people have a lot of trouble breastfeeding. A family member of mine was not able to, and people treated them terribly. It was awful. So with that, maybe they can't produce enough milk. Ducts and things just not working properly. So bottle feeding, especially with modern formulas, is pretty good. Like when we're talking about saying breastfeeding is slightly better, that statistically you can see a difference. If you take 400 children in an experiment, 200 bottle fed, 200 breastfed, on average, there's a slight difference for those that are breastfed. So that's if you were to compare that just by general visual or like test scores when they're 5 or something like that, you'd see essentially, you would probably observe no difference. But if you did a more rigorous test, you'd see a slight difference. With that being said, most studies aren't actually an experiment. It's probably not easy to get an experiment like that where you require some parents to breastfeed and require some parents to only bottle feed, and then controlling that along the whole entire experiment. So there's been very few of those, and usually they're with very small samples. So most of the studies of this are correlational. All right. I think that's enough detail in breast versus bottle feeding. If you want more information, this is actually a really good video on it. It goes into a lot more of the actual facts of it, so feel free. So with this, dynamic systems theory is a theory about the development of motor skills of children. Essentially, it says that infants develop their motor skills by perceiving and acting in their environment. So we see this little image basically showing some milestones at different ages. Obviously, that is on a range. So this is on average. That's about the milestone you'll hit that. Some people will be a little earlier developing. Some people will be a little later developing for some, like so many things in life, obviously. As babies try different motor skills out, those that work will stick around and will be improved on. Those that don't work won't stick. So that's part of the brain. A lot of that is the neurons in the brain, making connections, saying, oh, well, these are the neurons that have to be active for me to roll over. Oh, I'm trying to reach and grab this object. Oh, I did it successfully. That's a reinforcing thing. So that's a connection that's going to be stronger now in their brain and in their motor and sensory neurons. So the physical capacity and the neural capacity are both working together. So obviously a very, very young infant doesn't have the strength, the muscle capacity to say, reach out and lift up a block. They couldn't do that. So they need to build that muscle, that physical capacity, but it also takes time to build that neural capacity, to build those networks to be able to have the capacity to reach out, grab a block, and come and put it in your mouth. So with this, if a baby sees something as well, sees someone moving, sees someone, a parent playing with something, sees a sibling doing that, they will attempt to do similar kinds of movements. So kicking legs, moving arms, they'll try to mimic it. A lot of that's down to motor or mirror neurons, so neurons that activate when we observe someone doing something. It helps with us learning how to do actions. So if you've ever seen someone, let's say you're taking guitar for the first time, you watch your guitar teacher play a certain chord or play the strings in a certain way, your brain is actually firing through the neurons, the motor and sensory neurons at the same time. So you're kind of experiencing it while you're watching it. That's what a mirror neuron does. Yeah. So the main point here is don't just stick a baby in a seat or a crib and have it not moving. It's really important for the baby to be out, be moving, be exploring the world in whatever ways that they can. Playing with objects, hearing sounds, seeing different people's faces is a really important one as well. So reflexes. So these are inborn reactions to stimuli, just like our reflex to pull our hand back from a hot stove. These are other reflexes that had survival value, might even now still have survival value built into our evolution and our genetics. So the rooting reflex, you might have experienced this if you've ever been near a very newborn baby or a young baby and touching their head, they will move their head towards whatever touches their face and try to suck on that thing. So if you have Lego around, probably don't have that by the baby's face, or they'll try to suck on it. If it's a bottle or a pacifier, that would be a better thing, obviously. So, and that's called the rooting reflex. Obviously, that has survival value because it helps the child eat and feed and survive. So sucking reflex as well. If anything is in their mouth, they will suck on it. This is my favorite reflex. I'm not going to go into detail on all the reflexes. This is my favorite reflex. It's called the Moro reflex. When a young infant is startled by a noise or a movement, they will flail their arms and legs out really wide and try to grab onto anything that they can. It's my favorite because it's funny. And maybe I'm a terrible person, but I would sometimes go and just like spook my children when they were really little enough to have that Moro reflex, just boo or something like that, to watch their eyes go wide and their hands fling out. Hopefully I didn't traumatize them too badly, but the reason that's probably evolved into humans is probably from our pre-human ancestors. So when the pre-human ancestors were living in trees, if they fell or heard a loud noise, they might be falling out of a tree. And so spreading limbs probably enabled them to slow their fall, as well as the parents to grab onto a limb to save the child. So that's probably why that is an evolved reflex, and it's lost pretty early on in life. But another reflex is the reflex to hold a grip. That one similarly probably came from like hanging on to the parent as they were climbing through trees, which is pretty neat. All right, so in the time that we have, let's cover some gross motor skills. Gross motor versus fine motor. Gross motor are using large muscle groups, so like arms, chest, legs, versus fine motor skills are things using the dexterity in like your fingers. So with this, trial and error helps a lot for a child. As you can see on this little milestone, hopefully you can see it well enough, it's large enough to see. Grabbing a toy and rolling over, grabbing the toy is a really fine motor skill, but rolling over, that's a gross motor skill. The walking is a gross motor skill. Crawling, that's gross motor. And obviously this depends on the perceptual abilities. So being able to see and have a goal to like go over to the ball over there. If you can't see and perceive that, which very early on, your visual perception is pretty short and pretty blurry, but then by about 6 or 7 months, it's almost to 20/20, and then by about 9 months, it is at 20/20. So things are dependent on other systems. So the body and the development of the brain and the muscles and motor skills are all these systems working together is essentially the point I'm trying to make here. Oh, there, I made it bigger. So fine motor skills, grasping, so grasping a block, using a spoon, manipulating things, that takes a lot longer to develop. A lot of that because, one, you need the muscles to know what to do, and 2, you need the neurons to know what to do as well. So reaching and grasping are one of the first fine motor skills that appear, and they're really important for the development of the thoughts and abilities of a child to plan and do things. Up until they can really grab something and bring it to them, most of what's happening is just happening to them. They're really not choosing, hey, I want this thing. They might want to be fed, but that's really a human need, a reflex. But if they say, oh, I want this blue block versus this green block or something like that. So that's some of when the first parts of like making decisions can really be seen in a child. So this is a good video. We won't watch it in class right now based on time. It's observational learning based on mirror neurons. So it goes into a lot of depth on mirror neurons, not just what they do for observational learning, but also what they might do for development of empathy. So really interesting video, but we don't have time right now. So sensation and perception. I've used these words a couple times today. Hopefully you remember what they are from your book. But sensation is the messages that you're getting from your sensory receptors. So from your rods, your cones in your eyes, from your hair cells in your ears, from the chemical sensors in your tongue, or the pain, pressure, or touch receptors in your skin. That's the sensation. So we're getting information from the world in. That's a sensation. Perception is now, what do we do with that? What do we interpret that to be? The sensation is there is this frequency of sound, or there's this organization of sound. It's the actual sound data, you could say, coming into your brain. That's a sensation. Your perception is, oh, that's my favorite song. Obviously, for an infant, they probably don't have a favorite song yet, but is that a shape? Is that a color? Is that a pattern? Is that a car? Is that a dog? Those kinds of things. So the sensation is the data coming in. Perception is our interpretation. What is that thing? So how do we research infant sensation and perception? For an adult, you can ask them, hey, what do you see when you look at this? Do you see colors? Do you see a pattern? You can't do that with an infant, right? They don't have the language capacity yet. But it's really interesting how we can still see, how we as researchers can look and see what an infant's sensations and perceptions are. They can't report back to us what they hear, but we can look at their behaviors. So one thing that we use a lot is the visual preference method. Basically, we sit a child down. You might put a blue block and a green block. And if they are looking more at the blue block, that might suggest that that child is more interested in the blue block. You can also see what wavelengths can children see differently. So you put 2 different shades of blue. So you put 1 shade of blue and then a second shade of blue after they seem to get bored of that first shade of blue. See if they can tell a difference. Because children will get bored of a certain stimulus after a while. If you have this 1 shade of blue, they might find it interesting for a time, or a blue block. Maybe all the blocks are the same shade of blue, and then you put a new shade of blue in there. If they go for that, that might suggest that they can see that shade of blue as distinct from the others. So that's how we use the visual preference method with this habituation and dishabituation. So habituation is basically present the stimulus until they get bored with it, and then dishabituation is are they going to just be able to discriminate? Are they going to be able to tell that difference? So another thing that we can do is eye tracking. So you can have a eye tracking machine just put onto a child's head, which basically has cameras that look at what the child is looking at and at what their eye is. So it knows, oh, the child looked at this part of a screen for longer, then they went to this part of the screen, then they went to this part of the screen, then they went to this part of the screen. So you can look actually where a child is looking at, for how long, similar to that habituation, dishabituation. Put 2 different shades, see if when you change the shade of blue or something, if they then move their eyes, because people tend to look at changes in anything, color, light, things like that. So what do infants see? Again, we've learned this based on these kind of observational studies where we've looked at their behavior, look at what they prefer. So giving them different pictures of different faces, seeing if they can tell a distinction between them. So it seems like for 1 month old, this is about how well they see a face that's close to them. At 2 months old, about here, 3 months old, and then a year old. So at about 6 months, you're at 20/40 vision, which might be close to what my actual vision is right now. And then closer to 9 months, you're at about 20/20 vision. So, and then at a year, you pretty much, or you should, unless there's some problems, you should have definitely 20/20 vision by that time. And so you can imagine a 3-month-old that's trying to roll around and interact with things. What they're seeing is similar to this, right? So they can see faces, they can see a little bit further or a little more distinction. So presenting them with different objects, different shapes, different faces is really helpful for them to learn how to use their brain to interact with the world. So infants don't perceive occluded objects until after about the age of 2. So basically they learn that by experience, that when this object here, someone under 2 wouldn't necessarily be able to identify that as a cat, because it's like a couple different things. There's this part, and that looks like a cat, but then there's this line here, and then there's this other thing over here, and that looks like a cat. So yeah, our perceptual abilities, our sensations, we're getting the same, well, they're getting similar visual data, but then their perceptions of, oh, this is a cat behind something, are still developing, because they're experiencing, well, one object can pass in front or behind of another object. So depth perception, about 3 to 4 months old, so about when we're seeing at about this level, is when we start to see depth perception in infants. So 6-month-old is probably not going to crawl into the glass. They have clear enough vision to be able to detect that difference in depth. For a younger child, one, they're not experienced with, say, like falling down the stairs or falling off a couch, because they're hopefully not being exposed to a lot of stairs and like being on a couch or a bed unsupervised. So one, they're still visually trying to get that perception, and also just understanding, well, what is gravity? So a 6-year-old probably won't crawl out here onto the plexiglass, even if the mom's calling them. Younger than 3 months probably would just crawl straight towards, even though there's this visual cliff there. Yeah, this can be a funny video, a pretty good place to stop, especially with the end of class right now. So let's just watch this really quickly. We'll see in a couple examples of this as the last thing of class today.
In this study, babies between 9 and 12 months are brought into the lab and placed on a large plexiglass top table. Half of the table has a checkerboard pattern just underneath the surface, but halfway across is a visual cliff, which the baby can tell drops off steeply. The plexiglass top continues, so it's perfectly fine to proceed. But the baby isn't so sure, and this is a big drop for a baby just starting to crawl. She wants to get across to get the toy, but she's cautious and looks to the opposite end of the table where her mother is. The parent is instructed to smile or make a fear face.
If the mother is posing a fear face, the baby typically does not cross this stair step downward, this modified visual cliff or visual step. On the other hand, if the mother poses a smile or somehow poses a nonverbal communication that is not prohibitive but encouraging, the child is much more likely to cross over to her.
So there we're seeing the combination of socio-emotional factors as well. So this child, they're like, yep, I want that toy. Mom says it's cool. Let's go. Versus before, you saw those children like reaching out their feet as if they would if they knew they were going down a step. So at this point, this study is specifically being used to look at the socio-emotional connection between the mother and child, because naturally the child isn't going to want to do that because they have the sensation and perceptual ability at this point to see that visual cliff. But they're making the choice, the child's making the choice to go over because of this attachment and the encouragement from the parent. So we'll get more into socio-emotional development in infancy next time in class. So I hope everyone has a great weekend, and any other classes you have in between now and the weekend too. So yeah, thank you for being here today. If you've got any questions, I'll stick around for a few minutes and type them in the chat or use the microphone. Hopefully you learned a lot today about infant development. Thanks.